could I thank the organizers for this invitation and for getting the weather better than in Glasgow. Thank you. So my topic, I think, designed by Peter, was the sort of future. Omics, what's on the clinical horizon? So I thought I'll just be difficult and start from showing you a cover from Nature just a few months ago. I think it was when we were skiing in St. Moritz at the winter school that it appeared. And it tells us, I think, there is a little editorial to go with it. You'll see bits of it in a minute. That tells us that we use this word omics, or we add ohms to various bits of science that we do for an effect. It's rather negative. It says you just sort of dress things into omics to get money, basically, which perhaps we do. You'll tell me whether we do or we don't. So it divides the omics. This is, of course, a joke, but I think there is a bit of real thought put into it by one of the editors. So these are the sort of staff editors of Nature who write these things. So there are established ohms, and we know that those, and I'll talk about two of those, genome and proteome, but of course, we already heard in very good session this morning or early afternoon about transcriptome, and we will hear later about metabolome. They all established real. We know that there is a lot of data behind these ohms. But then people make up other things. And there are the emerging ones, such as variome. I'm not sure I subscribe to this one. Interactome, etc. Epigenome, however, is real, and we already know that there are lots of very interesting data in cardiovascular disease, particularly in cancer. And then there are aspiring ones, and those are perhaps shouldn't be there apart from phenome, that there is now big phenome consortium, as you know. So the next bit of the article, and this sort of figure talked about the good, the bad, and the ugly. Clearly, this audience doesn't need these instructions, but I thought it was quite funny how the criteria of being a good ohm differ from the criteria of bad ohm. I'm not going to read all this, but I particularly like the unpronounceable. So if you're unpronounceable ohm, then don't bother. It's not going to give you a big grant for millions of euros or pounds. So these are the jokes. The jokes are good, but what about the reality? So for a number of years, we'll be looking, and I like this way of showing it, from genomics to metabolomics, so from the code to the function, really, through the mechanisms, and we've added over the years microRNAs and many very interesting processes and mechanisms. But we still know a lot about genomes and genomics, and know less as we move down this pyramid. And I think that was clear during the earlier presentations. Now, just looking back a little, because you can't look into the future without looking back, the younger people in the audience probably don't remember, but some of us do remember that when we started, we did candidate gene association studies, and this was one marker, one polymorphism in one gene, low number of subjects, some patients, some controls, frequently below 100 in each group, and we were doing analysis and conclusions. And everything was very nice, and you could do it in one group during one PhD project, but the problem was that we couldn't get any reproducibility. So after some years of doing this, people stopped and started doing genome-wide linkage studies. And Mark Caulfield, who is here, many of us in the UK came together to do the BRIGHT study, which was published in The Lancet many years ago. And this was a classic linkage study. And this was good, and it was a little more reproducible, but you remember that areas under the linkage peaks were so huge that there were hundreds of thousands of genes there, And so-called dense mapping, Mark, was very, very difficult to do, that we promised in our paper. And then, of course, GWAS came, and you don't need talking me about GWAS, because we already heard about it a lot. And this was really a revolution, because we started seeing lots of things that mattered at very low p-values, highly significant, hugely interesting, and the whole industry of putting data together, meta-analyzing many of the studies developed. And it was great, but it's not the end of the story. And we heard in the first talk that we haven't yet explained everything, so that's good. We have another 10, maybe 20 years of fun with genomics, because we still have lots of normal variants and abnormalities and big abnormalities to explain. So that's good news for me. You know this, but I think even if you know it all, it's important to stop and look at it again, how complex it all is. We sometimes just show one Manhattan plot and one signal that was highly significant, but of course enormous amount of work that have gone to these studies is frequently forgotten. So your first discovery samples, your validation, your replication, and then taking it to functional studies, and I'm sure Mark Caulfield will talk about this later. Risk prediction, I think, is attractive. But whether it's ready for the clinic and whether we're doing any good by taking GWAS risk prediction to the clinic, I would be interested to discuss with this highly educated audience. People do it, but I sometimes worry. We'll see some good examples, and I sometimes worry whether we do any good or harm by doing this. Now, I'm sure Mark will tell you more because he was a senior author of this study, but I think that's, rather than showing you lots of GWAS for hypertension, this is the penultimate, the best, the biggest, the fantastic GWAS. So you remember this, published in Nature, September 2011. Lots of groups of patients from all over the world, gigantic work by Mark and colleagues and many groups around the world, and 70,000 individuals in 29 cohorts. You can't really do any bigger. That's it. So this is some more detail. You remember that there will be 27 SNPs targeted for validation, that lots of work has been done, that were 16 novel genome-wide significant loci. This is important because these frequently show completely new pathways, new mechanisms, things we haven't thought about before as in any way related to disease or trait we're interested in. But just going back to what I said about previous types of studies, this replication of previously published loci also hugely important. We've never had so much replication in any complex trait genomics of hypertension ever. So this was really something fantastic that happened, culminating many years of work and struggle and all the ACE genotyping that didn't work. So this was hugely important. This is familiar to you, Manhattan plot, a nicer Manhattan plot than majority of Manhattan plots because the p-values are very, very low. So the heights are very high here, as you can see. And there was a comment again from Nature editorial that findings presented in this paper could contribute to diagnostic and therapeutic studies. Could. Have they so far? Probably in some way. I am an optimist, but more work is needed. And it's that two Manhattan plots that show systolic and diastolic pressure that show why. Here you see the individual SNPs, red systolic, blue diastolic. The effects, as we all know, and we heard it already for other traits, are small. Systolic blood pressure, 1 millimeter mercury, or a bit less, or a bit more. Diastolic, 0.5 millimeter mercury. We know that if we give first drug to untreated patient with essential hypertension, we frequently see blood pressure lowering of 20, certainly 10 millimeters mercury. 1 millimeter doesn't sound very strong. Mark put in discussion a very nice sentence that from the point of view of epidemiology and complications, this 1 millimeter is important. And we know that it is important because that means many more strokes and many more heart attacks. But nevertheless, for individual patient I see in the clinic, 1-to-1 on Tuesday afternoon, that 1 millimeter mercury perhaps doesn't matter that much. Now, Dan Levy, who was the co-author of this study, led the risk score analysis, which was published together in the main paper, and he presented the data very nicely 2 years ago in Milan at the very nice genetic session that was when the data were just about to be published. And as you see here, what he did, he looked at this 29 SNPs in aggregate altogether, so you need to assume that you've inherited the bad bit of all 29s, and he could see that this score properly adjusted and everything was done possibly mathematically to do it as well as one could possibly, gave this very strong association with LVH, that's the top one, occurrence of stroke, the middle one, and coronary artery disease. And you see very, again, low p-values, 10 to minus 9 for CAD. That's very impressive. Interestingly, and we talked a little about it early on today, the renal abnormalities, the chronic kidney disease that we see so frequently in patients with essential hypertension didn't come out in this risk scores, and I'm not sure we still know why, and it's very interesting and requires more thinking. So we have something. There were many other papers afterwards. This is just one of them, and this is Schunkert. We've seen already some of his data in the first presentation. This is now looking at a very large cohort of CAD cases, more than 20,000 cases and more than 60,000 controls, and looking at these blood pressure SNPs, all of them, and looking how that contributes. So as we heard before, there are pleiotropic effects. The same SNPs are important in more than one trait, or maybe simply having hypertension throughout many years is obviously predisposing you to coronary artery disease. But these are very strong effects, again, slightly differently shown. These are not p-values, but equally strong effects. So there is something in it, but whether we would be rushing to the clinic to tell our patients that your risk of stroke is X and how this would help us, I think not yet, probably. Sandosh mentioned studying extremes, and I am not apologizing for mentioning uromodulin again, because good things need to be repeated frequently. So this is a collaboration, of course, with Ulla Melander and colleagues here in Malmö. And I think it illustrates beautifully that if you have good patient and controls, you can go far. And this is mentioned previously, Malmö Diet and Cancer Study, and these are the hyper controls that Sandosh described in detail. And this was important because it allowed us to probe from extremes and find something that other studies didn't find, even with 70,000 people. These are the data. So this was the most extreme GWAS ever for hypertension. It's never been done before, and it will never be done again because no one has patients like that. And even if you would start gathering them now, it would take a while to gather such patients. So, and particularly controls, the most difficult were controls. You have seen this Manhattan plot already. Not staggering, nothing as tall as Mark's 70,000 people, but very interesting. And very interesting because if we added number of other studies pre-selected for extremes, it has become hugely significant at 10 to minus 13, but now the numbers are much larger, almost 40,000. But even more importantly, it sits in the gene, and as you know, very few other SNPs that came significant in many of GWASs actually sit definitely within the gene, and this one is beyond any doubts in there. So this was interesting. Sanders already presented a number of studies that followed. I'll just summarize what the UMOD or TAM horseful protein or uromodulin is. So this is the gene, the protein been known for years, and yet no one could put any function to it for many, many years. And the work that Sanders and colleagues are now doing, the functional work, will fill this gap. It's very large, untidy, glycosylated protein. When we went to our crystallographers and said, could you do a crystals for us and look at the structure, they laughed because it's such huge and untidy protein to work with. But the beauty is that it is where salt is and where important kidney hypertension functions happen. That is, it's tubular protein, but in humans is expressed primarily or almost uniquely in the TAL, in the thick ascending limb of the loop of Henle. So that tells us already that we're into something really interesting. And I won't repeat everything, just the summary. I think the beauty of this is that the functional studies that we've done and put into the original paper showed us that this is a protective event, that perhaps we are now searching in a new pathway that protects against rise of blood pressure throughout life, throughout many years. And that's nice, because what we need in the future as a community are new drugs that affect new mechanisms, because the pipelines of new antihypertensives are completely dry, and that affects the way the whole area and the research in our area is progressing, because there is fantastic research on both sides of Atlantic. There are fantastic groups that do wonderful functional studies, but drug industry hasn't got interest in what we come up with. And we need to bring them back, because we need some new drugs. We haven't yet solved all the problems of hypertension. So just to summarize that a bit, that's how we've been. We've done candidate genes, wasn't great. We've done genome-wide linkage. We did well, but not yet answered things. We did GWAS. This was a huge progress, but we're not yet there. There is lots of this unexplained variability. There are still genetic determinants that we haven't explained. So what's next? We have seen some beautiful examples with the diet and genes just a few minutes ago on gene-environment interaction. There is a lot to do in this area. There is a lot to do on gene-gene interaction, but I think that next-generation sequencing will show us the missing heritability, will discover this middle variant on the well-known slide of looking for the missing variability. But it also might do some other things, and these other things, I believe, are the broad area of stratified medicine. And this picture, you might like it or not, is designed to show you that personalized or stratified medicine, I must say I prefer the word stratified medicine, because personalized gives people and industry a very wrong impression that you want to have a different drug for every individual, a different test for every individual. And that makes our industry partners run away from us further than you can say hello. If we say stratified medicine, they like it more, because we are dealing in cardiovascular medicine with very common traits and diseases, and therefore there are millions or maybe billions of people they can treat, and that becomes, from business point of view, a good proposition. So stratified medicine, I think, rather than personalized. I think the areas of pharmacogenomics, risk stratification, we already covered, drug efficacy, dosing, and side effect prediction. So all the stuff in this corner is very interesting and ready to start on to work on. And there are some very good examples in cancer sciences, for example, because they already there and we can follow if we clever. So we know already, and Sanders alluded to this, there are well-established genetic determinants of response to some cardiovascular drugs, and warfarin would be a fantastic example where FDA already requires American physicians to do the test. Not the same is not true in Europe, but probably will become so. There are a number of prospective trials, and Sanders alluded to some of them, that show this very nicely. And the question is, when would we translate to the clinic near you? Not easy, probably not for every drug, but there are some very interesting examples and opportunities there. And this is an example of relatively recent study from Mayo Clinic, Stephen Turner. He's done quite a lot of nice work in this area. And here you see that mirror image of angiotensin receptor blocker, candesartan, and classic thiazide diuretic hydrochloroside. And you see that for this one genotype, this is just one example, there are others, as you would expect, you see opposing almost mirror image type of responses because, of course, candesartan does well in those subjects with very active renin-angiotensin system, and hydrochlorothiazide does well in those where volume is the driving factor. You would say we can measure renin. Well, very soon it will be much cheaper to do a whole genome than to measure renin. So these methods might be pretty good for clinical practice at some point. Now, what we have taken from all this in Glasgow is that one shouldn't do it separately for every disease, but rather we should have a collaborative effort to try to tackle all chronic diseases because the baseline information you need, the technology is the same. So what we've done is we put together all chronic disease issues related to genomics, and we call this Stratified Medicine Scotland Innovation Centre. We raised £20 million to start this work, and we have a number of industrial partners, some real, and those are in the middle here, the Life Technologies, the sequencing company, and Aridia Informatics, which is big health informatics company, which actually put their own money and in-kind contributions. Others watch whether we succeed and might join later. So clearly, we've already heard that sequencing is getting cheaper, cost per genome is getting less. And you see here that it will go very quickly or is almost close to $1,000. And then it becomes a real proposition that we can do it as a business proposition. So the company that we work with acquired a new technology, very expensive, but very efficient, better technology than previous technologies. This is visualized here on the back of the Mini Metro. It is portable. It can be done in every laboratory, in every clinic, really. It traveled across UK, and we've had several of our students having their genomes sequenced in our courtyard, really, in the university. But seriously, doing things like that, doing many genomes sequenced in patients with hypertension, allows this big data activity, allows us to join, to link the next generation sequencing with real-time patient data, of course anonymized, and that's what we're preparing to do. We haven't done yet, but we're preparing machinery to do this. It also teaches us really to work in that network of information and brings together business, clinicians, and people who have the technology. So I think it's very positive. What we aim to do is to do clinical trials with big pharma in a new way. You see these drug trials get smarter heading. So rather than, including everybody in the drug trial of one drug or a combined medication, and then doing a test and then selecting those who respond and rejecting everybody else, you can do it in a real time. You can have your data from either limited gene panel or entire genome and select to 4 trials at the same time without wasting time. And that's the idea, that's the hope, that that's how it's going to work. Of course, I mentioned that cancer physicians have been there already. They're already doing it in some lethal cancers. And the best example is probably pancreatic cancer. And that's where my examples are. So if you have a tumor, such as pancreatic cancer, where survival is 6 months, there is enormous push for the clinician not to waste time. And there is a paper just after Christmas in Nature where our colleagues contributed. And there is huge international collaboration on pancreatic cancer where this is being done, where stratified medicine is actually in the clinic, in practice. And I think we can do the same in cardiovascular disease. There is really no inhibitors to do so. We just need to come together and be clever about it. So I just wanted to give you another omics, because we can't just stay with genomics after all these cartoons at the beginning. So proteomics is the one that we've been developing for about a decade in Glasgow, and it's interesting because it has different features than the genome, of course. You remember that, like genomics, proteomics tries to do the whole proteome to understand everything. That's very ambitious because that's the function of all the cells and all the organisms together. But you can have some shortcuts, and for us, urinary proteomics, which is really peptidomics of fixed urinary set of hundreds of peptides in the urine sample, has been a shortcut to understand some insight into proteomics. And this is the platform that has been established by colleagues in Glasgow, and it allows you to look at thousands of these peptides at the same time very quickly. 45 minutes is normally enough to look at hundreds of peptides that describe the pattern using bioinformatics. And for example, we published a number of papers of detection and functional changes in evaluation of coronary artery disease, and you would think that urinary peptides and coronary artery disease don't link, but they do. Very well controlled, very well tested studies where you see dynamic changes, whether with exercise or, as on the next slide, drug therapy. And you see here that if you take patients with both hypertension and coronary artery disease and give them a short period of treatment with angiotensin receptor blocker, nothing happens. But if you give the same angiotensin receptor blocker for 2 years, after the 2 years, there's been some remodeling of the coronary vasculature and other vasculature, kidney vasculature, and you see the good response. You see that bioinformatic measure that describes all the hundreds of peptides. Sometimes these are 300, 400 together. There is a beneficial effect. And this shows this dynamic character of proteomics, which of course is more difficult to show in genomic investigation. This is chronic kidney disease. You remember in some of our genomic risk, we couldn't really combine our SNPs with chronic kidney disease. Here we can. So many common polypeptides are abnormal in patients with hypertension, coronary artery disease, and chronic kidney disease patterns, because this is probably all vascular disease that is then shown as through the kidney in this polypeptide bioinformatic final analysis. So I'm almost there. I just wanted to show you now all the omics together, because I think really the beauty is that you can add all of them together. And I see more and more papers that, despite the cost, try to do at least 3 out of 4. And this obviously becomes more and more logical because the flow of information The flow of information is here, yeah? So if you want to use the omics for mechanistic studies, it would be nice to have genetic, transcriptomic, proteomic, and metabolomic data on the same large group of well-characterized patients. It's still difficult to do, but I'm sure we'll see that soon. So Peter wanted me to predict the future. I think it's very difficult to predict the future, and I thought Saint-Exupéry said it very nicely. We just need to work hard to enable the future rather than try to guess things that cannot be guessed. I wanted to acknowledge people who do the real work and various funders of ours, and particularly EU Mascara, which is a nice collaboration between many labs all over Europe that looks at biomarkers of cardiovascular risk. And last but not least, I would like to remind you that we are just before a very important meeting in Milan that will do lots of things, thousands of reasons you see to attend. But one very important one is, of course, that new guidelines, 2013 ESH-ESC guidelines will be revealed to the world, and it's very soon. And if you haven't yet decided to come, you still have the time to come. So please join us in Milan. Thank you for your attention.